what are you? Not who, but what. It seems like a simple question. Science should have a definition of life, but it doesn't, at least not a working one. The best attempt is NASA's 1994 formulation. Life is a self-sustaining chemical system capable of evolution. It sounds solid, until you try to apply it. Fire sustains itself, spreads, and adapts to its environment, but no one would call it alive. A mule breathes, thinks, and feels. It's obviously alive, but it's sterile, doesn't reproduce, so by definition it doesn't qualify. A virus completely breaks the system. Outside a cell, it is inert like a crystal. Inside, it takes over and replicates. We cannot give a clear answer even to the basic question of alive or not. Yet everything else depends on it. We send robots to search for life on other planets, but we cannot define exactly what we're looking for. We're trying to recreate in the lab the moment when chemistry became biology. 70 years of attempts, thousands of experiments, 0 results. And the deeper we dig, the worse it gets. Because if you look at life from the outside, it looks like an infection. It covers every surface of the planet, penetrates every crack, mutates, spreads, and seizes resources. And some serious scientists seriously believe that this is exactly what it is. Something that infected Earth from deep space. Every answer makes the question deeper. And this is only the beginning. Let's look at what physics says about you. You are made of 7 octillion atoms, mostly carbon, hydrogen, oxygen, and nitrogen. But here is the thing. These are the exact same atoms you can find in dirt, in rust, or in ash. There is nothing special about the atoms themselves, and they all obey the exact same rules of the universe. The biggest rule of them all is the second law of thermodynamics. To put it simply, everything in the universe is constantly moving toward disorder. Things break down, heat escapes. Over time, structure falls apart until nothing useful is left. In physics, there are no exceptions to this rule, except whatever is happening inside you right now. Back in 1943, the famous physicist Erwin Schrödinger pointed out a massive mystery. He asked a question that the science of his time couldn't answer. How do living things resist this universal law? How does a random collection of ordinary atoms manage to build, maintain, and repair itself, while everything else around it is falling apart? Schrödinger wrote a famous book about this called What is Life? It was so powerful that it inspired the scientists who later discovered DNA. But here is what that book does not contain. The actual answer. Schrödinger said that life survives by essentially feeding on order to fight off the chaos, but he couldn't explain how that engine actually works, and honestly nobody who followed him could either. Sure, today we can map chemical reactions. We have fancy names for every molecule, but we still don't completely understand why your specific pile of atoms holds together and stays alive, while the exact same atoms sitting on a table do not. Think about it. If that mysterious organizing engine inside you suddenly switched off, not slowly, but instantly, the bonds holding your cells together would just stop working. In a matter of seconds, your body would revert back to being just basic chemistry, just like the dirt beneath your feet. I think about this whenever I hear people say that we have biology completely figured out. We really don't. What we have is a list of results. We are watching a magic trick and writing down what happens, but we don't know how the trick is actually performed. We don't have control. We just have a really good vocabulary to describe what we see. So, what exactly is this invisible force that reverses disorder inside a living cell? Why does it only work in biological matter, but not in regular matter made of the exact same stuff? And if we don't even understand the very foundation of life, what does that mean for everything else we think we know? You might say, well, even if we don't understand the mechanics, we can at least recognize life when we see it, right? But in 1935, Even that basic assumption was frozen in its tracks. Or, more accurately, it was crystallized. Right off the back of that mystery, in 1935, a scientist named Wendell Stanley did something that broke the rules. He took a living pathogen, a virus, and turned it into a crystal. I don't mean that as a metaphor. I mean actual geometric crystals. Static, lifeless, exactly like the structures you see in minerals or table salt. He extracted the tobacco mosaic virus from an infected plant, crystallized it, and put it on a shelf. Later, he dissolved those crystals back into water. And the virus was still active. It was still invading cells. It was still doing exactly what biology says a living thing is supposed to do. Stanley won the Nobel Prize in Chemistry for this in 1946. But the massive question his experiment opened up has never been closed. Here is why this is so mind-bending. A crystal has no metabolism. It has no internal engine running, no digestion, no chemical activity of any kind. It is basically a rock. And yet, here was infectious biological material existing in that exact same geometric stillness, doing absolutely nothing, going nowhere. It was effectively paused until the environment around it changed. And when it did, the virus simply hit play and resumed exactly where it left off. Traditionally, biology puts everything into two neat boxes, alive and not alive. Stanley demonstrated that these two boxes are missing a massive grey area. And here is where this stops being a story about plants and starts being about you. Your body's immune system identifies threats by looking for biological activity. It scans for chemical signals or the noise of an invader trying to replicate. But something that has completely suspended its own biology produces zero signals. To your immune system, a crystallized virus reads as dust, just a harmless mineral particle. It drifts through your body's entire heavy-duty detection system without triggering a single alarm. And it's not because the virus is doing some sophisticated stealth maneuver, it's because it literally has nothing to hide. It is essentially dead matter. It arrives as an inert object, and it only becomes a problem after it is already inside the vault. A suspended pathogen reaches your soft tissue. It finds the perfect moisture, the right temperature, and the cellular machinery it needs. Then, it wakes up. The invasion spreads outward through your tissue, and you are fully alive while this happens, completely unaware. There was no alarm at the gates because alarms require biological activity to trigger. The enemy had no activity until it had already breached the walls. Honestly, I find this more disturbing than almost anything else in this field. Not because it's some sci-fi theory, but because it is fully documented, awarded the highest scientific recognition available, and the terrifying category problem it exposed was never actually fixed. So, what else exists in that dark space between alive and not alive? What else can hit pause, travel undetected through your bloodstream, and resume operations deep inside you? What is our real defense against something that pretends to be simple chemistry and only becomes a biological threat after it arrives? Well, the next problem is even worse. Stanley's virus was a zombie, sure, but it still had genetic code. It still needed a living cell to hijack. But what happens when the invader needs absolutely nothing? No DNA, no RNA, no cellular structure at all, and brings your entire system down anyway? Back in 1958, Francis Crick laid down the absolute golden rule of molecular biology, known as the central dogma. It goes like this: DNA makes RNA. RNA makes protein. It is a strict one-way street. Proteins are the end of the line. They are just the building blocks. They cannot replicate themselves, and they absolutely cannot, on their own, spread a biological threat. Then came 1982. A scientist named Stanley Prusiner published evidence that, actually, a protein could do exactly that. The scientific community didn't just politely doubt him, they rejected him completely. But he was right. And it won him the Nobel Prize in 1997. Enter the prion. A prion is simply a misfolded protein. Think of it like this. Every protein in your body is a chain of amino acids folded into a very specific three-dimensional shape, like complex molecular origami. That exact shape determines its job. But a prion takes an alternative shape. It becomes chemically stable, but functionally completely wrong. And here is the terrifying part. When this rogue prion bumps into a normal, correctly folded protein of the same type, it forces the normal one to change its shape and copy the mistake. Then, that newly flipped protein does the exact same thing to the next one, and the next. It's a silent domino effect moving through your tissue. It does not stop, and your body's defense system registers absolutely nothing. When this chain reaction hits brain tissue, the structure simply breaks down, leaving tiny gaps everywhere until the brain essentially looks like a sponge. Now, how do you stop it? You don't. Standard sterilization does not work. Superheated steam, the method they use to clean surgical instruments, does nothing. Bleach? No. Boiling it? No. Formaldehyde? Radiation? Useless. Why? Because you cannot break a shape. Prions have no metabolism to disrupt. They have no genetic material to damage. They are just a folded piece of protein, and that fold survives literally everything we throw at it to eliminate every other known threat. Ordinary water can carry prions. They can slip right through standard filtration systems, and they reach you. This is the exact moment where I stop thinking of biology as a neat system we actually understand. Instead, we are just living inside a house without a complete set of blueprints. A completely lifeless object, with zero genetic code, enters your tissue and starts converting it. And the body doesn't notice a thing until the structural damage is permanent and irreversible. If a biological takeover does not even require genetic material, what exactly is driving the process? What else spreads this way that we haven't even isolated or named yet? And where is the line between basic chemistry and a full-scale threat, if crossing that line doesn't even require DNA? But somehow, the next layer of this mystery goes even deeper. A prion, as terrifying as it is, at least needs your proteins to do its work. It uses the materials already sitting inside you. So, what happens when an invading agent needs no existing biological material at all? If a prion is essentially a physical shape manipulating other shapes, what happens when an invader brings absolutely no physical tools of its own? What happens when the threat is nothing but pure, naked information? Think of a living cell like an incredibly advanced automated factory. It is brilliant, but it has one catastrophic blind spot. The machinery inside cannot distinguish between its own internal instructions and random blueprints that just drifted in from the outside. That is not a unique flaw in one specific animal or plant. It is a fundamental hardware vulnerability in how life operates. And in 1971, a scientist named Theodore Diener published a paper detailing the smallest entity ever found to exploit this vulnerability completely. For decades, researchers had been trying to figure out what was silently devastating potato crops around the world. Under electron microscopes, they found no bacteria, no protein coats, no classic viral shapes. What Diener finally found was just a floating, naked loop of RNA, between 246 and 375 nucleotides. No shell, no armor, just an exposed string of genetic code. He called it a viroid. A viroid carries zero equipment. It has no enzymes, no molecular tools, no engine. But when it slips into a cell, it hijacks the cell's own enzymatic infrastructure to copy itself. The cell reads the viroid's RNA and just runs it. It doesn't check the source, it doesn't ask for an administrative password, it just executes the command. Imagine barely 250 letters of code issuing orders inside a massive, complex living factory, and having those orders followed without a single question. And here is the absolute genius of its geometry. Why a circular loop? In biology, your defense enzymes act like security guards. They destroy rogue linear RNA by grabbing onto its exposed loose ends and chewing them up. But a closed circle has no loose ends. It is structurally seamless. Because of that, it resists breaking down. It survives longer in the wild, travels further, and reaches deeper into living tissue before anything can even try to stop it. If it reaches a cell with the right environment, the cell simply runs the loop. It is foreign code redirecting your cellular machinery to produce something your body was never designed to make. And there is no built-in firewall to stop the process once it begins. I keep returning to the terrifying simplicity of it. You can target complex biological threats because they have lots of moving parts. But this is just a line of instruction meeting a machine that follows instructions blindly. It makes you wonder, what is the actual inventory of functional, circular RNA structures floating in the air of a busy city over a full year? What is the absolute minimum snippet of code required to completely redirect human cellular processes? And if our internal machinery cannot verify the source of the code, it runs. What exactly is your cell executing at this exact moment? But that question becomes infinitely darker when you look at the code that is already permanently written into your DNA. Instructions that aren't drifting in from the outside today, but were hardwired into your sequence millions of years before you were even born. To understand how foreign code ended up permanently hardwired into you, we first have to look at how biology actually shares information. For the longest time, the concept of a species assumed a strict, secure genetic boundary. We thought of it like a closed system. You have a shared genome, you pass it down to your offspring, and it only changes through slow, random mutations over many generations. This classic idea of a family tree was the absolute foundation of how we understood biological identity. But in 1977, a scientist named Carl Woese published a paper that completely shattered that foundation. By sequencing RNA across hundreds of organisms, Woese discovered an entirely hidden third domain of life called archaea. It had been completely invisible to the neat, two-category system biology had been using for a century. But more importantly, this discovery, later expanded by Lynn Margulis in 1981, proved the existence of a mechanism connecting all these branches of life. It's called horizontal gene transfer. Put simply, bacteria do not just inherit traits vertically from their parents. They can take genes directly from their neighbors. It doesn't require reproduction. It requires just one single contact event. It is essentially genetic airdrop. In a matter of minutes, a piece of genetic code can pass between two organisms of completely different evolutionary lineages. Take E. coli, for example. It lives in your gut. It lives in soil. It is in standing water and on almost every surface that has ever touched organic material. And it is constantly acquiring and donating genetic material. Resistance to extreme radiation, the ability to produce new chemical defenses, resistance to literally every class of antibiotics currently used in modern hospitals. These traits are not waiting to evolve. These genes already exist in environmental bacteria right now. They are completely available, and the file sharing network is highly active. Some people argue that for a superbug to become resistant to multiple drugs, it would require a highly unlikely series of simultaneous mutations. But Woese and Margulis's work tells us a much darker truth. These transfers are not rare. They are continuous. The genome of literally every single bacterium you currently carry inside your body is at least partially made up of borrowed code from completely different, unrelated lineages. I keep returning to what this actually means for modern medicine. When we create a new antibiotic, it targets a specific molecular structure. But the fix to beat that antibiotic, the resistance gene, already exists somewhere in the global bacterial population. We are not racing against slow, random mutation. We are racing against an open source library that is already complete, just waiting for the right bacteria to download the update. How many unknown resistance genes are floating around in soil and water, just waiting to be transferred to clinical pathogens. What does this genetic file sharing look like in deep-sea microbiomes we have never even sampled? And if every bacterial genome is basically a patchwork of borrowed code, what does biological identity even mean? And more importantly, what does it mean for us? Because this ancient mechanism of downloading foreign code didn't stop with single cells. It happened to your ancestors. And the proof is sitting right there in the sequence you were born with. Code that wasn't borrowed during your lifetime, but was incorporated millions of years before the human species even existed. Remember that ancient downloaded code I just mentioned? Here is the exact number. 8% of your entire genome is not actually human. It belongs to ancient retroviruses that infected your evolutionary ancestors anywhere from 40 to 100 million years ago. They didn't just infect a random skin or lung cell. They invaded reproductive cells. They wrote their own sequence directly into the heritable code, and they never left. When the Human Genome Project was completed, scientists found these ancient viral ghosts scattered across literally every single chromosome. For most of the 20th century, biologists confidently called this junk DNA. They thought it was just broken, inert evolutionary clutter, doing absolutely nothing. They were completely wrong. Take a sequence called HERVW, a human endogenous retrovirus. This ancient viral code is actively expressed in the human placenta. The protein it produces is structurally essential for creating the cellular layer that allows nutrients to pass between a mother and a fetus. Without that specific viral protein, that interface cannot form. And without that interface, mammalian live birth is physically impossible. Think about that. You exist right now because an ancient infection permanently hacked your ancestors' reproductive biology millions of years ago. The body didn't clear the infection. It built its entire operating system around it. The virus became a load-bearing pillar of human life. And the rest of these ancient sequences are not all silent either. Another one, called HERVK, actually produces detectable viral particles in human embryos during early development. We still don't fully understand what they are doing during that window. Other viral sequences now act as management, controlling the volume on human genes that have nothing to do with the original virus. It is completely surreal to realize that 8% of the instruction manual defining your biology was written by a blind process that had absolutely zero interest in your survival. It only cared about copying itself. And now, you structurally depend on it. Normally, your body has heavy suppression mechanisms, molecular locks, keeping the most dangerous of these viral sequences permanently silenced. But those locks are not unbreakable. Accumulated DNA damage puts immense pressure on those mechanisms. If a repair system makes a mistake, a lock can fail. Suddenly, you have a partially functional viral sequence waking up and running wild inside tissue that was never built to handle it. This is exactly why extreme environments are so terrifying. Take deep space travel, for example. The intense radiation of space continuously compounds DNA damage over months. No one has fully mapped what that kind of sustained damage will do to the molecular locks holding back the ancient viruses sleeping inside living human tissue. What is the full biological profile of these ancient codes? What exact threshold of stress disrupts their suppression? And what else in that 8% have we confidently labeled as harmless junk, simply because we haven't yet seen the conditions that trigger it? Our viral past is invisible to us because it literally became us. But the next massive blind spot in biology is invisible for a completely different, almost impossible reason. It's a threat that is built on the wrong hand. At the end of the last part, I mentioned a threat that is invisible simply because it is built on the wrong hand. Here is exactly what that means. Hold your two hands out in front of you. They are made of the exact same components, but they are mirror images. You cannot perfectly superimpose your left hand over your right. Molecules work the exact same way. They have a geometry, a handedness, known as chirality. Back in 1848, Louis Pasteur was looking at crystals under a microscope and discovered something that still haunts biology today. He found that life had made an absolute universal choice. Every single amino acid, in every single protein, in every living organism on this entire planet is left-handed. They are called A form. The right-handed mirror versions, D amino acids, are chemically identical. They are made of the exact same atoms, but geometrically, they are completely reversed. And for some reason, biology totally excludes them. No one knows why. After more than 170 years, we just have guesses. But the result is a massive, structural blind spot. Think of your enzymes and your immune system like a highly specific left-handed glove. A right-handed molecule simply does not fit. So, what happens when your body encounters a D molecule? The enzyme just moves on as if nothing is there. Your immune system scans it, but the molecular patterns don't register. The entire right-handed half of molecular reality is not flagged as a threat. It isn't flagged as food, either. It is just invisible, processed as inert background noise. But here is the problem. If a D-form compound gets into your tissue, it doesn't get metabolized, and it doesn't get flushed out. It just accumulates. Your cells constantly bump into it, try to process it, fail silently, and move on. The recognition alarm that tells your body, hey, we need to destroy this foreign material, is never triggered. The material just sits there. It is the biological equivalent of undeletable junk files, slowly filling up a hard drive. Imagine multiplying that effect across all your tissue, across years and years of breathing in uncatalogued particles from modern environments that contain D-form compounds. This isn't a sudden, violent event. It is a silent, creeping accumulation inside tissue that your body is desperately trying to maintain while your internal software completely fails to flag that anything is wrong. 4 billion years ago, life made a total geometric choice. And that choice created a total blind spot that covers literally half of all possible molecular structures in the universe. It is not a software bug we can patch. It is the permanent cost of biology's original decision. So, what exactly is the D amino acid content of the air we breathe in a crowded city over a full year? What does a decade of accumulating undeletable, right-handed molecules actually do to human tissue? And if our foundational biology ignores 50% of molecular reality, what has already slipped through that blind spot? Well, in November 2024, 38 scientists published a massive warning about exactly what happens when something steps into that blind spot all at once. Harvard, MIT Cambridge, the Max Planck Institute. In November 2024, 38 top researchers, synthetic biologists and biosecurity specialists, published an open letter in the journal Science. It wasn't a standard request for more funding or research. It was a massive red flag. A warning to humanity to stop before we even start. The subject of the letter? Mirror life. We just talked about the right-handed blind spot in chemistry. Now, imagine using that invisible material to construct a fully functional microorganism. The complete geometric inverse of all terrestrial biology. This isn't just a genetically modified organism. This is an entire parallel operating system of life, built on the opposite geometry from everything that has ever lived on this planet. The authors stated clearly that creating these organisms is technically achievable in the very near future, and they warned that their existence would be completely unmanageable. Here is why. A mirror-bacterium still needs to eat. It replicates. It competes with normal life for basic raw materials, like carbon, nitrogen, and phosphorus. Those basic elements don't have a left or right hand. They are up for grabs by any organism capable of using them. But remember that left-handed glove we talked about? A mirror organism has literally 0 left-handed parts. It presents no standard surface patterns. It has no recognizable chemical ID tags that your cellular guards are calibrated to scan. It doesn't trigger your immune system. Not because it is using some advanced stealth technology to hide, but because the detection system was simply never built to see it. It exists completely outside the biological dictionary your body uses to define reality. Because your body doesn't see it, it sends no signal to stop it. It just keeps replicating. The scientists chose their language extremely carefully. They didn't just say mirror life would be dangerous, they said it would be uncontainable. If you face a bacterial threat today, we use antibiotics. But an antibiotic works like a physical key fitting into a specific lock. In a mirror cell, that lock is reversed. The antibiotic has absolutely nothing to bind to. Every single medical countermeasure humanity has ever invented operates strictly in left space. Mirror life operates completely outside of it. Honestly, I find the cold, clinical precision of this warning much more disturbing than a general panic. These experts identified the exact gap, the exact competition for resources, and the terrifying absence of any response toolkit. And they concluded that once that threshold is crossed, there is no known way to put the genie back in the bottle. What kind of rules even exist right now to govern research into complete mirror inversion? If this dropped into an open environment, how long would it take to ruin a local ecosystem? And given that every alarm system you possess is built for the other hand, how would you even know if it had already happened? But while we worry about synthetic mirror life escaping a lab in the future, the next problem is already sitting inside you. It's not arriving from the outside. It's already running, hardwired into your sequence from the moment of your very first cell division. I told you the next threat was built into your sequence from the moment of your very first cell division. Here is exactly what that means. Every single cell in your body is counting. And I don't mean that metaphorically. At the very end of every chromosome, there is a physical countdown clock. It's a repeating sequence of DNA called a telomere. Think of it like the little plastic tip at the end of a shoelace that keeps it from fraying. Every time your cell divides, that protective tip gets clipped just a little bit shorter. When it is finally gone, the chromosome begins to degrade. The cell simply stops dividing. A scientist named Leonard Hayflick discovered this back in 1961. Before him, the absolute scientific consensus was that human cells in a lab could divide forever as long as they were given enough nutrients. People thought aging was just accumulated damage from the environment. Hayflick's data ended that assumption completely. He proved that normal human cells divide approximately 50 times, and then they stop. The environment can be perfect, the food can be there, but the cells just cease. It's a hard-coded stop sign now known as the Hayflick limit. But here is the mystery. Evolution does not keep expensive, complex mechanisms around unless they serve a major purpose. Having a built-in self-destruct counter actively limits your own lifespan. Why do it? The standard explanation is that it's a defense mechanism, a way to stop old, error-filled cells from mutating into dangerous, unstoppable rogue growths, and that is plausible, but it doesn't explain the extreme precision. A blunt defense mechanism doesn't stop at exactly 50 divisions every single time. Something inside you is counting to a very specific number. And we still don't know who or what set that number. It also doesn't explain why some cells completely ignore the limit. Your reproductive cells, for example, express an enzyme called telomerase. It acts like a biological repair crew, constantly rebuilding the ends of those shoelaces. Because of this, they can divide forever without constraint. But you know what else does that? The most aggressive, dangerous, unstoppable cellular mutations in human biology. They flip that exact same switch to become immortal. And we still don't fully understand how that switch works. We do know what makes the clock tick faster though. Radiation accelerates it. Chronic inflammation accelerates it. When astronaut Scott Kelly returned from a year on the International Space Station, he had measurable, persistent changes in 7% of his genetic expression. The counter runs entirely differently under the extreme conditions of space, a place humanity is now actively trying to move into. I honestly don't think we have seriously reckoned with what it means to carry a biological lifespan defined by a literal countdown timer. A timer with an original setting we didn't choose, and one we cannot read directly. What exactly determines whether a cell turns that immortal repair enzyme back on under stress? Why does that exact same enzyme save a reproductive cell, but turn another cell into an unstoppable biological nightmare? And if this countdown limit evolved to protect us from one specific failure mode, what kind of unseen failures is the limit itself creating? To keep that cellular countdown clock ticking, your body requires a constant stream of energy. And to understand how energy actually enters the living world, we have to look at a process that, according to the standard rules of physics, simply shouldn't work as well as it does: photosynthesis. According to classical physics, when a plant absorbs sunlight, that particle of energy should bounce around randomly inside the cell like a pinball. It should lose most of its power as heat long before it ever reaches the plant's engine. And yet, plants convert sunlight into chemical energy with a level of perfection that makes our most advanced, human-engineered solar panels look like a joke. How? In 2007, a team led by Graham Fleming at UC Berkeley published a mind-bending answer in Nature. The particle of light doesn't just bounce down one random path. It uses a loophole called quantum superposition. It travels every possible path simultaneously and instantly collapses onto the single, most optimal, perfect route. This means that life is literally running a quantum computation inside every single green leaf, every algae, and every photosynthetic bacterium in the ocean, continuously, at room temperature. This quantum trick is exactly how the raw energy of the universe enters the living world. But here is the terrifying catch. Quantum coherence, this delicate balancing act, only exists under very specific fragile conditions: the right temperature, the right electromagnetic environment, the precise geometry of molecules. These conditions are not permanent, fixed constants. They fluctuate. The physics holding your biological machinery together isn't actually a solid, indestructible system. It is just a temporary, localized configuration of conditions that currently happens to be producing order. Remember in the very first part, when I asked what would happen if the engine fighting entropy suddenly failed? This is the physical reality of that. If your local environmental conditions shift by even a fraction of a percent in the wrong direction, the delicate quantum effects keeping your chemistry afloat would degrade. Your cells would suddenly struggle to extract energy from food. The electrochemical signals keeping your neurons firing would collapse. In seconds, the bonds organizing your tissue would break down, fading back into the disorganized chemistry of everything around you. People love to say that the laws of physics are stable, but what we really mean when we say that is, they just haven't visibly shifted during the extremely brief window humanity has existed to measure them. That is a completely different claim. What exact range of electromagnetic shifts is capable of disrupting this delicate quantum balance in active biological systems? And if the foundation of our core chemistry is this precisely calibrated, exactly how razor-thin is the margin keeping us here right now? We depend entirely on this fragile quantum dance with sunlight. But the next question is about what happens when you take all of that away. No sunlight, no quantum solar magic, no surface at all. Just life, buried in complete geological isolation for 1 billion years, surviving on an alien chemistry we have barely begun to map. At the end of the last part, I asked what happens to life when you take away the sunlight and bury it deep underground for 1 billion years. Well, when we actually look down there, the answer sounds almost impossible. The total mass of living organisms currently sitting beneath your feet is greater than the total mass of every human, animal, plant, and insect on the surface combined. And not just by a little bit. In 1992, a scientist named Thomas Gold proposed this exact idea, and the scientific community completely rejected it. People thought he was crazy. But later, drilling data from the Deep Carbon Observatory proved that the drill bits didn't lie. They found microbial life extending up to 5 kilometers deep into solid rock. These are bacteria and archaea living in trapped geological water, in absolute pitch-black darkness, under crushing pressure, and at temperatures that would instantly melt a standard lab's petri dish. These organisms have never seen sunlight. They do not run on our biological clock. Some of them metabolize so incredibly slowly that they might only complete a single cell division once every few thousand years. It is like an engine idling so slowly you can barely tell it is running. They are biology operating on geological time. They are alive by every measurable standard, but they are completely disconnected from the biosphere we know. In fact, Earth essentially has two biospheres. The one we study, the one you see outside your window, the one every rule of biology is built upon, is just a paper-thin film on the surface. Beneath it is a vastly larger, heavier operating system running on entirely different chemistry and different rules, and we have sampled practically zero of it. But here is where it gets concerning. Modern deep drilling operations are actively piercing into formations that have been securely sealed for millions of years. When we open those vaults, we introduce surface conditions to organisms that have never encountered them. More importantly, we introduce what was sealed down there to the surface. Earlier, I mentioned how your immune system can be blind to threats with the wrong geometry. This is a completely different kind of blind spot. Deep microbes have absolutely zero evolutionary history of coexistence with surface biology. Your immune system was shaped exclusively to fight threats that evolved above ground. Neither side has any preparation or any defense protocols for making contact with the other. When scientists publish papers describing these deep biosphere samples, I keep noticing the word uncharacterized. They use it constantly, and it isn't just a temporary placeholder waiting to be filled, it is a confession. We have no idea what we're looking at. What is the actual chemical profile of a microbial community that has been sealed 3 kilometers deep inside solid rock? What exactly happens to human tissue upon contact with an organism that evolved in a completely separate alien reality? And if the mass of the hidden biosphere massively outweighs the life on the surface, which biosphere is actually the anomaly? Them or us? The extreme crushing conditions that shape that deep biosphere are actually a massive clue, because they point directly to the place where life on Earth most likely began in the first place. Not in a warm, sunny pond on the surface, but in extreme heat, extreme pressure, and toxic chemical gradients at the very bottom of the ocean. Conditions that are still actively churning out there right now. Imagine the very bottom of the ocean. A place where absolutely zero sunlight has reached in the entire history of the Earth. The pressure down there is 300 times what you feel on the surface. Near certain geological fault lines, the water temperature exceeds 400 degrees Celsius. On the surface, that water would instantly explode into steam, but down here, the crushing pressure keeps it liquid. By all known logic, nothing should be able to survive this. But in 1977, a submersible called Alvin descended into the pitch-black Galapagos Rift and found something that completely broke biology. They found entire thriving ecosystems clustered around hydrothermal vents. Cracks in the ocean floor shooting superheated, toxic, mineral-rich water into the freezing deep. There were tube worms 3 meters long, giant clams, and blind shrimp. And they weren't running on sunlight. They were running on chemosynthesis. The bacteria there were eating the raw chemical energy of the Earth itself. Since the 1990s, scientists like Nick Lane and Michael Russell have pointed out something incredible. These alien-looking vents, they aren't exotic exceptions to the rule. They are the original rule. Life on this planet most likely began in exactly these environments. These mineral chimneys generate natural electrical charges, specifically proton gradients. That sounds like complex chemistry, but here is the mind-blowing part. It is the exact same chemical battery system operating inside your own cells right now. The microscopic engines powering your heartbeat today are direct descendants of the chemistry bubbling in those vents. The origin of life isn't just an event in the past. It is an engine that is still running, at hundreds of locations on the ocean floor right now. But here is the terrifying constraint. We just talked about drilling into solid rock on land, but the formations beneath these active ocean vents, the sealed rock chambers that have been brewing organic material since before complex life even existed, are largely untouched. They have been sealed off from our surface world for millions of years. What happens when a deep sea drill accidentally breaks one of those seals? Physics takes over. Whatever is pressurized behind that seal instantly shoots upward into the water column. And unlike solid rock, the ocean is not a barrier. It is a massive, high-speed distribution network. A single microscopic agent released at that depth gets caught in deep-sea currents that connect every single ocean basin on the planet. An unknown replicating code released at the bottom reaches every coastline, every fishery, and every organism relying on the ocean in a matter of years. It's a timeline that makes any organized global response functionally impossible. I am not writing science fiction about what that agent might be. I am simply pointing at the massive, terrifying gap between what is currently locked in those deep-sea formations and what we actually understand. What containment protocols even exist for deep ocean drilling in these geologically active zones? Are those safety rules built to handle threats we know, or threats we can't even imagine? And if the exact extreme conditions that originally assembled the very first living molecules are still running at the bottom of the ocean today, what is currently assembling in the dark, just waiting for us to crack the seal open? We just saw how deep-sea vents act like giant chemical batteries, but that brings us to a mind-bending realization about biology itself. Life doesn't actually need food. It just needs electrons. When you eat, digest, or breathe, your body is essentially just doing complex math to move electrons between molecules. But in 2012, researchers in Denmark found something that cuts out the middleman entirely. Deep in marine sediment, they discovered cable bacteria. These microbes link together to form literal living wires, up to several centimeters long. One end sits in oxygen-rich water, the other deep in sulfide-rich mud. They don't ingest anything. They don't digest anything. They just pull electrons from the mud and pass them up the chain. They are running a biological electrical circuit directly through their own bodies. Now, take that logic one terrifying step further. A microbe that eats electrons doesn't know the difference between natural mud and a buried copper wire. An electron is an electron. The source is entirely irrelevant. This means an organism capable of drawing current from its natural environment is also perfectly capable of drawing current from human infrastructure. Think about the power grids, underground internet cables, and server arrays buried right now under our major cities. These bacteria don't need a geological source, they just need an available current. I am not talking about some sci-fi biological attack. I am talking about an entirely natural appetite. These organisms already exist. They already live adjacent to our buried infrastructure. What exactly happens to the global power grid if our copper cables get covered in a biofilm of organisms that literally eat electricity? If biology evolved the ability to consume electrical current millions of years before humanity even invented the light bulb, why do we confidently assume our underground networks are safe? But somehow, the next organism goes even further. It doesn't need a copper wire, and it doesn't need a chemical battery. It feeds on pure, deadly radiation, and the higher the dose, the better it grows. At the end of the last part, I mentioned an organism that feeds on radiation. Here is exactly what that means. A lethal radiation dose for a human is about 500 rads. To completely sterilize a medical lab, you need a few thousand. But back in 1956, scientists tried to sterilize canned meat with an absolute nuke of a dose. It spoiled anyway. Inside, they found a bacterium called Deinococcus radiodurans. It had just survived 1.5 million rads. At that extreme level, radiation doesn't just damage DNA, it completely shatters it into hundreds of tiny, disconnected fragments. It is the biological equivalent of putting a fully built Lego set into a blender. But within 3 to 6 hours, this microbe casually reassembles its entire genome. This is where the physical mechanics become truly baffling. We know some of the proteins involved in the repair, but we completely lack a physical explanation of how the cell maintains the precise positional information of its code. When DNA is shattered, the pieces should drift apart into chaos, but somehow, this organism manages to keep hundreds of separate fragments suspended in the exact correct spatial arrangement, even after the structural backbone of the DNA itself has been destroyed. It puts the puzzle back together with absolute precision, and the cell simply resumes normal activity. But here is the deeply unsettling mystery. Evolution is a strict economy. It does not build complex, biologically expensive armor without a massive environmental pressure forcing it to do so. Yet, environments producing 1.5 million rads do not naturally occur on Earth's surface. Nuclear reactors didn't exist millions of years ago. So, what exactly was this organism fighting in its evolutionary past that required this absurd level of protection? Deep space transit? Asteroid impacts? We literally have no verified answer. Now, imagine dropping a similar extremophile into an active nuclear reactor today. It doesn't experience the core as a threat. It experiences it as a food source. And what exactly is keeping that radioactive fissile material safely separated from the outside world? Shielding and coolant systems, metal, polymer, and concrete. These are basic materials that microbial biofilms have possessed the chemical capacity to degrade since long before humans existed. The combination of an appetite for radiation and the ability to slowly eat through industrial materials creates an outcome that standard engineering specifications were never built to handle. If this organism evolved the ultimate defense long before humanity even invented the weapon, what other alien capabilities are waiting out there for conditions we haven't even built yet? Well, the next problem isn't about extreme survival. It's about the life forms we are completely blind to right now, simply because we built all of our instruments to only look for what we already know. At the end of the last part, I said our instruments are completely blind to anything we aren't already looking for. Here is exactly how big that blind spot is. Every single instrument we use to detect life makes one massive assumption: that life uses the exact same chemistry we do. Our tools don't actually detect life. They detect familiarity. In 2005, a scientist named Paul Davies formalized this terrifying logical gap. He argued that if life started more than once on Earth, a completely independent second genesis using a different chemical formula, its descendants could be sitting right in front of us, and every laboratory on the planet would systematically fail to detect them. He called it the shadow biosphere. This isn't science fiction, it's basic probability. The raw ingredients for abiogenesis, chemical precursors, thermal energy gradients, and reactive mineral surfaces were not unique to one single moment in Earth's history. Those exact conditions recurred constantly. If they successfully sparked life once, there is absolutely no reason to assume a completely independent second genesis would use our exact same chemical alphabet. Remember the mirror life we talked about earlier? If a shadow organism naturally used those reversed amino acids, it would simply starve in a standard lab petri dish, because our culture media are strictly built to feed our specific biochemistry. And our DNA sequences are hardwired to only read our standard 4-letter code. If they scan an organism using completely different nucleotides, or a metabolism running on unrecognized chemical substrates, the machine doesn't sound an alarm to announce an alien discovery. It just registers it as broken data, as static. This means every soil, water, and air sample humanity has ever collected was processed through filters designed to ignore anything that doesn't look like us. Whatever percentage of those samples consisted of chemistry our instruments couldn't recognize was simply thrown away, labeled as background noise or contamination. The shadow biosphere, if it exists, has been systematically filtered out of every dataset we have ever produced. For decades, science has treated the phrases, "it does not exist," and "our instruments are not designed to find it," as the exact same statement. They are fundamentally not. If a shadow biosphere has been quietly coexisting alongside us for billions of years, what is it doing with the resources it has been quietly consuming? And more importantly, what happens when a biological operating system completely alien to ours decides to interact with us? While we wonder what shadow biology might be hiding right next to us, another completely alien threat is waking up from the ice. The permafrost is not a graveyard. It is a freezer. In 2014, scientists extracted a virus from Siberian permafrost that had been sealed for 30,000 years. When they thawed it, it immediately began hijacking amoeba cells. The virus, Pythovirus sibericum, wasn't weakened at all. It was functionally identical to the day it was frozen. And 30,000 years is just the shallow end. Deeper layers hold biological material frozen millions of years before modern mammals even existed. Just like the deep rock microbes we discussed earlier, this ancient material has absolutely no shared evolutionary history with our modern immune system. Our bodies have zero defense protocols for it. Right now, millions of square kilometers of Arctic terrain are rapidly thawing. Material sealed for eons is washing into rivers and oceans. Some of it will be inactive, but some will not. For example, deep permafrost contains bacteria that produce carbonate, the exact same biomineralization process that builds seashells and bone. If that microbe wakes up inside your respiratory tissue, it doesn't just stop. The chemistry keeps running. It can literally crystallize soft tissue within hours. You are fully awake while this happens, and there is absolutely no external off switch. Everyone discussing permafrost thaw worries about methane and carbon. Almost no one pays attention to the ancient biological archive waking up alongside it, and the thaw is not waiting for our research to catch up. What exactly is the viable biological inventory of permafrost sitting 15 meters below the Arctic? What happens when ancient proteins make contact with modern tissue they have never encountered? What the ice preserves through freezing, another environment preserves through absolute geological isolation. And that next environment is not frozen. It is highly active. Lightning plus water plus 1 week produces the building blocks of life. In 1953, Stanley Miller demonstrated this at the University of Chicago. He took a sealed flask of basic chemicals, water, methane, ammonia, and hydrogen, and ran a continuous electrical spark through it. 7 days later, 5 of the essential amino acids used in every living protein on Earth appeared without any biological assistance. In 2008, scientists reanalyzed Miller's preserved samples with modern equipment. They found 25 amino acids, literally every building block required for protein chemistry, assembled from non-biological scratch and a single spark. People treat that experiment as a neat historical milestone. But the conditions Miller used are not just laboratory artifacts. They are ongoing geochemical realities. Think about volcanic lightning over sulfur-rich emissions, or electrical discharge during massive storms over heavily polluted, warm industrial cities. The exact same experiment is running continuously right now, at a massive scale, in the sky right above us. It has been running since before life existed, and it didn't stop when life first appeared. And the chemical yield of this ongoing experiment is not zero. The novel amino acids it constantly creates are highly reactive. They fall with the rain, contact our skin, and enter our respiratory tracts with every breath. But here's the problem. Remember when we talked about those invisible, wrong-handed molecules silently accumulating in your tissue like undeletable junk files? The exact same silent failure applies here. Because these are completely novel chemical configurations that our enzymes were never shaped to process, our biology doesn't know what to do with them. The body tries to run the chemistry, fails to complete the step, and simply registers the failure as background noise, Over years of continuous exposure, these unrecognized reactive compounds accumulate and actively interfere with cellular repair at the molecular level. The surface of your skin is just the first point of contact. It is not the last. Our medical tests only measure what is catalogued against known biological standards. What is uncatalogued produces absolutely no signal. I find the distance between how often the Miller-Urey experiment is cited in textbooks and how rarely its continuous, present-tense operation is taken seriously to be one of the strangest blind spots in modern biology. What exactly is the complete chemical yield of a massive thunderstorm over a polluted industrial zone? And if the exact same spark that assembled the original precursors of life is running continuously over every major city today, what is it actually producing right now? Well, the molecule that takes those raw precursors and turns them into a self-replicating machine is already inside you. It predates DNA. It predates proteins. And it still runs the core machinery of every single cell you have. Your cells contain the literal molecular fossil of the moment life began. Before DNA, before proteins, before anything we would recognize as biology, there was RNA. In 1986, Walter Gilbert proposed the RNA world hypothesis. His core argument was brilliant. RNA is the only known molecule capable of both storing genetic data and physically acting like a machine at the exact same time. The very first self-replicating molecule didn't need DNA for instructions or proteins for action. It only needed itself. And that ancient machinery is still running inside you. The ribosome, the microscopic factory that builds every single protein in your body, is fundamentally just an ancient RNA catalyst. This fossil isn't preserved in rock. It is fully functional, operating inside you right now. Remember the viroid we talked about earlier? That naked loop of foreign code that slipped into a cell and hijacked it? This ancient fossil is exactly why your biology is so wildly vulnerable to it. Your ribosomes evolved billions of years before complex biological threats existed, which means they have zero built-in verification. They operate with total, blind trust. If a self-replicating fragment of foreign RNA reaches your lung tissue, your ribosomes don't ask for a password. They just read the code and start building it. The cell is quietly hijacked into producing copies of something that only cares about its own replication. There is no dramatic biological alarm. The tissue just silently redirects its energy. And that foreign fragment doesn't even need to arrive fully formed. RNA precursors naturally concentrate and link together on common mineral surfaces, like clay or iron sulfide. These minerals exist in everyday soil and in the dust suspended in the air we breathe. The jump from scattered chemicals in urban dust to a self-replicating genetic sequence is entirely possible. The conditions are not rare. It is deeply disturbing to realize that the most ancient, foundational factory inside your body, the factory that literally makes you, will blindly manufacture absolutely anything it is handed. If these RNA fragments are actively assembling themselves in the dust of our cities, what is the absolute minimum sequence length required to silently hijack a human lung? And if your internal factory cannot distinguish its own instructions from foreign code, what exactly is it building right now that didn't originate inside you? Life is not a random accident. It is a phase transition. The distinction here is incredibly important. An accident is a miraculous sequence of unlikely events converging once in a billion years. But a phase transition is inevitable. Think about water. It doesn't accidentally freeze. If the temperature drops low enough, freezing is simply what water does. The question isn't if it happens. The question is when. In 1993, a scientist named Stuart Kauffman formalized the mathematics behind this. He proved that if you have a dense, diverse enough chemical soup, the probability of molecules building each other crosses a mathematical threshold. Molecule A builds molecule B, B builds C, and eventually C loops back and builds A. The system closes on itself. It becomes self-sustaining. No DNA required. No cells required, just chemistry crossing a mathematical line. And Kauffman's models suggest this threshold is shockingly low. You don't need a primordial ocean or a deep sea vent. You just need a concentrated mixture of organic compounds. Now think about urban wastewater infrastructure. The sewer systems beneath our major cities are massive, churning, enclosed rivers of chemical and biological diversity. They mix the output of millions of humans, industries, and atmospheric runoff. The molecular diversity down there exceeds Kauffman's required threshold by orders of magnitude. And the chemistry doesn't know it is inside a pipe. It only obeys the math. If a brand new, self-sustaining chemical loop spontaneously emerged in our sewers tomorrow, it wouldn't announce itself. It would just produce a little heat and consume organic matter, blending in perfectly with the normal bacteria already living down there. But there is absolutely no rule constraining this loop to produce something harmless to us. Lifeless chemistry spontaneously rebooting itself into a new biological system right under our streets isn't science fiction. It is a mathematical prediction based on conditions that exist right now. But while Kauffman's transition happens completely spontaneously in the dark, the next terrifying threshold required human hands, and we have already crossed it. In the last part, we saw how lifeless chemistry could spontaneously reboot itself. But in March 2016, human hands forced a reboot inside a laboratory. Craig Venter and his team published a paper on a synthetic organism called JCVI-SYN 3.0. Their goal was incredibly ambitious. They wanted to find the minimal genome, the absolute smallest set of instructions required to sustain life. Starting with a known bacterium, they systematically stripped away genes until the organism stopped functioning, then added back only what was strictly necessary. The final result had just 473 genes. It lives, it divides, and it passes its genome to the next generation. But here is the deeply unsettling part. Out of those 473 essential genes, exactly 149 have a completely unknown function. Let that sink in. We stripped life down to its absolute core, to the bare minimum required to exist, and we still have no idea what nearly a third of that code actually does. Biology absolutely requires those instructions to live, but humanity cannot read them. Remember when we talked about ancient permafrost microbes having no natural predators? This synthetic organism is infinitely worse. Every naturally occurring bacterium on Earth is kept in check by a massive ecological immune system, natural predators, bacteriophages, and competing organisms that co-evolved over millions of years. JCVI-SYN 3.0 exists completely outside that web. It has no ecological relationships whatsoever. If a containment failure happens, we aren't just releasing a known pathogen into an environment with established defenses. We are releasing an organism that no ecosystem has ever processed into every ecosystem simultaneously. It would spread through water, through air, through contact with literally any organic material it encounters. And its reactions with uncontrolled environmental chemistry are not predicted by any existing model, because 149 of its foundational instructions have unknown outputs. A synthetic organism consuming organic material in an open environment doesn't stop at a natural boundary. It stops when the food runs out or when something physical stops it. I have looked at the biosafety assessments for synthetic biology. Their risk categories are built entirely around the behavior of known organisms in known environments. The entire safety framework relies on one massive assumption: that you actually know what the organism does. Remove that assumption, and the framework simply has no applicable category to protect us. But while we worry about synthetic biology escaping a lab, the next threshold is moving entirely outside our planet. It is surviving conditions in the cold vacuum of space that should be absolutely lethal, proving that the boundary between Earth's biology and everything outside it is far more permeable than we ever thought. 10 days in open space. Vacuum, radiation, temperature swings between plus 150 and minus 150 degrees Celsius. Tardigrades returned alive. Ingemar Jonsson and colleagues sent tardigrades aboard Photon M3 in 2007. Full unshielded space exposure, 10 days, then rehydration, and viable offspring. Published Current Biology, 2008. Not ambiguous. Complex multicellular animals survived direct space exposure and remained reproductively intact. What the experiment established is transit capability. An organism inside ejected rock survives launch forces, vacuum, radiation, and re-entry across timescales required for transfer between adjacent planetary bodies. Every large impact in geological history ejected material at escape velocity. Some from Earth fell on Mars. Some from Mars fell here. The transfer has run in both directions for 4 billion years. In 1996, David McKay and colleagues at NASA published analysis of Martian meteorite ALH8 4001 in Science. Inside, carbonate globules, polycyclic aromatic hydrocarbons, magnetite crystals, and structures 20 to 100 nanometers in size. The paper proposed biological origin. The response was immediate and global. President Clinton addressed the nation. The question has not been officially closed. As of 2024, no definitive abiotic explanation for the full combination of features has been produced. The meteorite is in a museum collection. It has been handled, sampled, breathed around. If viable material transferred from Mars to Earth inside rock, some of it arrived before we were looking. Some arrived after. We did not establish a sterile baseline before beginning sample collection. We do not know what we introduced into laboratories alongside the rock. Martian material brought into warm, metabolically active environments finds conditions precisely opposite to the cold vacuum, where it spent millions of years. If anything inside was waiting, it is no longer waiting. I think about the specific sequence. We found the rock. We brought it inside. We argued about what it contained. We never seriously asked what happened to the environment we brought it into. What is the viable biological inventory of Martian meteorites currently in terrestrial collections? What sterile protocols governed sample handling in 1996? And were those protocols built around known threats or unknown ones? And if transfer between Mars and Earth has been running for 4 billion years, what arrived long before we start collecting? In September 2020, Jane Greaves and colleagues published a detection of phosphine in Venus's atmosphere at an altitude of 53 to 60 kilometers. Concentration, 20 parts per billion. Published Nature Astronomy. No known abiotic process produces phosphine at that concentration in those conditions. On Earth, phosphine is produced by anaerobic bacteria and industrial chemistry. Neither exists on Venus. No alternative explanation has been confirmed. Follow-up observations in 2021 detected the signal again. The altitude matters. At 53 kilometers, Venus's atmosphere has temperatures and pressures close to Earth's surface. It is the most Earth-like environment in the solar system outside Earth. If life exists there, it lives in sulfuric acid clouds. It breathes chemistry that destroys every material we use for spacecraft. A probe designed to sample that environment and return would carry whatever it found directly into Earth's atmosphere during a re-entry. Cassini detected something different in 2017. Hunter Waite and colleagues published in Science, Enceladus, Moon of Saturn, vents molecular hydrogen, carbon dioxide, methane, and complex organics from geysers breaking through its ice shell. The conditions at the base of those geysers are chemically identical to Earth's hydrothermal vents. The environment where life most likely originated on this planet. The same chemistry, the same temperature gradients, the same mineral surfaces. What lives in Earth's hydrothermal vents is not gentle. It is adapted to pressure, heat, and chemical extremes that would destroy surface organisms instantly. Whatever lives in Enceladus's ocean, if anything does, evolved in complete isolation from every organism on Earth. It shares no evolutionary history with our immune systems. Our immune systems share no history with it. We are planning to send probes to both environments. We are planning to return samples. I do not think the return protocols are built around the correct threat model. They are built around what we know. The threat is what we do not know. What containment protocol handles a sample from an environment that may contain life sharing no biochemical history with Earth biology? What does re-entry contamination look like for an organism adapted to sulfuric acid clouds? And if Enceladus's ocean has been running the same chemistry as Earth's origin environment for billions of years, what has that chemistry produced in isolation? Titan has lakes, an atmosphere, a full weather cycle with precipitation, evaporation, and river channels carved into its surface. The liquid is methane. The temperature is minus 179 degrees Celsius. There is no water. James Stevenson and colleagues published in Science Advances in 2015 a theoretical framework for cell membranes built from nitrogen-bearing organic molecules that function in liquid methane at Titan's temperatures. Not water-based, not carbon-oxygen chemistry. A completely different biochemical foundation that is thermodynamically stable in conditions that would instantly destroy every organism on Earth. We search for life by searching for water. The instruments aboard every mission to Titan were designed around water-based biochemistry. If life on Titan uses methane as its solvent and nitrogen compounds as its structural molecules, every instrument we sent was looking at the wrong signal. We have not found life on Titan. We have confirmed that our instruments do not detect non-aqueous biology. The Rosetta mission reached Comet Churyumov-Gerasimenko in 2014. The Kozak spectrometer identified glycine, phosphorus, and over 400 distinct organic molecules in the comet's material. Published Science, 2015. Earth passes through the debris trail of Comet Swift-Tuttle every August. For 2 weeks annually, the planet moves through material shed by a body that has traversed the outer solar system for billions of years. The particles enter the upper atmosphere. They descend. They land on skin and enter respiratory tissue through normal inhalation. What they contain beyond catalogued organics is characterized only for molecules our instruments were designed to detect. Instruments designed before anyone confirmed 400 organic compounds in cometary material at this density. Every August, the same debris field, for the entirety of human evolutionary history. The contact is not hypothetical, it is annual and ongoing. We have been inhaling material from the outer solar system for as long as we have existed, and we catalogue the contents after the fact, with instruments built for familiar chemistry. What is the biological activity profile of cometary particulate reaching the lower atmosphere during peak meteor shower periods? What does prolonged inhalation of outer solar system organic material do to human tissue over decades? And if we are searching for life only where water exists, what have we already dismissed as background noise? 40,000 tons of cosmic material fall on Earth every year. Matthew Murphy and colleagues confirmed this in Earth and Planetary Science letters in 2021. Not rock fragments visible to the eye. Fine particulate. Microscopic structures settling continuously into oceans, soil, and lungs. The material contains complex organics, nucleobases, and amino acids. It has been falling since before life existed. It falls now. Inhaled microstructures from space are not metabolically inert by default. Some are chemically reactive with biological tissue. Some are physically crystalline at scales that interact directly with cellular structures. The body processes what it recognizes. What it does not recognize accumulates. Microstructures from outside the Earth's biosphere have no entry in any biological recognition system, shaped by evolution on this planet. They pass through, they stay. The accumulation is not an event. It is continuous. Roberto Orose and colleagues published radar data from the Mars Express mission in Science in July 2018. The MARSIS instrument detected a reflective layer beneath the southern polar ice cap, consistent with liquid water. Liquid water under pressure in contact with mineral substrate at stable temperatures. The same conditions that produced life in Earth's deep subsurface. Proposed drilling missions to reach that layer would breach a sealed geological formation that has had no contact with surface conditions for millions of years. What is pressurized behind that seal moves toward lower pressure when the seal breaks. The drilling equipment returns to Earth. The sample return capsule enters Earth's atmosphere. Whatever was sealed beneath Martian ice, under pressure, for millions of years, arrives in the same biosphere as everyone reading this. Drill casing corrodes. Sample containers are not infinitely sealed. The organisms that would emerge from a pressurized Martian subsurface reservoir did not evolve in contact with any material we use for containment. They evolved in contact with Martian rock and Martian brine. The difference is not trivial. What is the reactive profile of cosmic microstructures in human respiratory tissue over years of continuous exposure? What containment protocol handles a pressurized subsurface Martian sample breaching its container inside a return capsule? And if we drill into a sealed Martian reservoir, what are we assuming about what comes back? Scott Kelly spent a year on the International Space Station. Frida Bailey-Jackson and colleagues published the analysis in Science in April 2019. 7% of Kelly's gene expression showed persistent changes after return. Telomere dynamics altered, cognitive performance shifted, gut microbiome restructured. His identical twin, Mark, on the ground, showed none of it. The ISS sits within Earth's partial magnetic shielding. A transit to Mars removes that shielding entirely for 6 to 9 months each direction. The radiation environment of deep space does not damage DNA gradually and uniformly. It breaks double-stranded DNA at random positions, including positions that regulate the suppression of endogenous retroviral sequences. The 8% of the human genome written by ancient viral infections and currently held silent by suppressor mechanisms. A suppressor silenced by a radiation-induced break means a partially functional viral sequence begins expressing in tissue not designed to handle it. The expression produces particles. What those particles do in the confined environment of a spacecraft, circulating among a crew with no medical infrastructure beyond what they carried from Earth, is not a scenario any mission profile has fully characterized. The crew returns changed in ways the pre-mission baseline did not predict. They return to a planet whose biology has no preparation for what they are carrying back. Every instrument we send to other worlds was built to find what we already know. The detectors read familiar molecular signatures. The assays test for known compounds. A planet with clean air and no detectable pathogens by our instruments is cleared for surface contact. The astronauts remove their helmets. The dust on that surface contains crystalline structures formed in chemistry we did not include in our detection protocols. The crystals enter lung tissue. They are physically sharp at the scale of cells. They do not trigger biological recognition because they are not biological. They are mineral. They tear. The lungs fill slowly with what the instruments said was not there, because the instruments were not looking for it. I do not think mission safety protocols have caught up with what we now know about non-biological chemical threats in extraterrestrial environments. What is the full range of gene expression changes in humans exposed to deep space radiation over 9 months? What crystalline mineral structures in Martian or lunar dust interact destructively with human alveolar tissue? And if our instruments are calibrated to find familiar threats, what are we cleared to walk into? Fred Hoyle calculated the probability of a functional enzyme assembling by chance from available amino acids. The result? 10 to the power of negative 40,000. Not small, not unlikely. mathematically indistinguishable from impossible within the time and volume available. Hoyle and Chandra Wickramasinghe published the conclusion in Evolution from Space in 1981. Life did not originate on Earth. The chemistry required is too specific, the probability too low, the timescale too short. Life arrived. The universe distributes biological material through space continuously, in comets, in interstellar dust, in material ejected from impact events across billions of years of planetary history. If Hoyle is right, life on Earth is not native. It is a colony. Whatever seeded this planet seeded others. The same material falling on Earth fell on every rocky body in every star system in the galaxy for 10 billion years. Life does not begin rarely. It arrives everywhere the conditions allow it to persist, which means the galaxy is not empty, which means the silence we hear from it is something else entirely. In November 2024, 38 researchers from Harvard, MIT, Cambridge, and the Max Planck Institute published an open letter in Science. The subject was mirror life, fully mirror-image microorganisms built from D amino acids, L sugars, and right-handed nucleic acids. The complete geometric inverse of all terrestrial biology. They did not say it would be dangerous. They said it would be uncontainable. Your immune system reads molecular patterns shaped around L geometry. A mirror bacterium presents no L amino acid surface structures as antigens. No cell wall patterns that pattern recognition receptors are calibrated to find. It competes for the same carbon, nitrogen, and phosphorus as every organism on the planet. It replicates. It does not stop. Nothing currently in the human pharmacological or immunological arsenal was built for the other hand. The 38 scientists asked that the research stop before starting. The letter acknowledged the research had not stopped. If life arrived from space, what else arrived with it that we have not yet identified as non-native? What regulatory framework currently governs research on complete chirality inversion in living organisms? And if mirror life were introduced into an open environment today, how would you know? DNA requires phosphorus. RNA requires phosphorus. ATP, the molecule transferring energy in every living cell, requires phosphorus. Without phosphorus, the molecular machinery of life cannot be assembled or maintained. There is no biological substitute. Isaac Asimov identified the problem in Scientific American in 1959. Phosphate rock is a finite geological resource. Dana Cordell and colleagues quantified it in Global Environmental Change in 2009. Known economically recoverable reserves, 50 to 400 years depending on consumption rate and new discovery. When the reserves are gone, they are gone. Phosphorus cycles through ecosystems but disperses into ocean sediment faster than geological processes return it to accessible form. Agriculture requires phosphorus. Without phosphorus, crops fail. Without crops, every complex organism at the top of the food chain loses its base. The bones soften first. Then the failure cascades upward through every system that depends on phosphate chemistry for structural integrity. Life on this planet is running on a non-renewable element and has no contingency for its absence. Fred Hoyle discovered a different constraint in 1953. Calculating nuclear resonance states in stellar fusion, he found that carbon-12 has a resonance at 7.6565 mega-electronvolts. Without this resonance, carbon does not form in stars. Without carbon, organic chemistry does not exist. Without organic chemistry, life as understood by every definition we have produced is impossible everywhere in the universe. The resonance value is what it is. A deviation of 4% in either direction eliminates carbon from the universe entirely. Hoyle stated publicly that a universe producing this resonance by accident looked to him like a universe that had been arranged. He did not retract the statement. The physics holding your biochemistry together is calibrated to a precision that has no physical explanation. The element your biochemistry requires to function is running out. Both constraints are absolute. Neither has a workaround. How much phosphorus remains in economically recoverable form under current agricultural consumption trajectories? What is the physical explanation for the carbon-12 resonance value and its apparent fine-tuning? And if the universe is calibrated for life with a precision we cannot explain, what does that precision imply about what the calibration is for? Robin Hanson published the argument at Harvard in 1998. If the universe is old, vast, and governed by chemistry that produces life wherever conditions allow, the sky should be full of signals. It is not. Something eliminates civilizations before they become visible. He called it the Great Filter. The filter is either behind us or ahead of us. If behind, we pass through it. The difficult step, the improbable origin, the rare evolutionary transition, we survived it and others did not. We are genuinely unusual. If ahead, the filter is waiting. Every civilization that reaches our stage of development encounters something that ends it. Technology itself may be the mechanism. The development of biotechnology, of artificial intelligence, of the ability to engineer self-replicating systems, these may be the final steps before the filter closes. Nick Bostrom stated in MIT Technology Review in 2008 that discovering microbial life on Mars would be the worst news in the history of humanity. Not because Martian microbes are dangerous, because it would mean life originates easily, which means the filter is not in the origin, which means it is ahead, which means it is coming. Svante Pääbo's team at the Max Planck Institute sequenced the Neanderthal genome, published in Science in 2010, for which Pääbo received the Nobel Prize in 2022. The Neanderthal genome contains the same endogenous retroviral sequences as the human genome. The same ancient viral infections that rewrote our ancestors rewrote theirs. The same 8% that is not ours is also not theirs. We share a viral inheritance with a species we outlasted. Those sequences are still active in both genomes. HERVK produces particles in human embryonic tissue. The same sequences in Neanderthal tissue produce the same particles. The virus that wrote itself into the ancestor of both species is still running in the descendant of one. The Neanderthals are gone. We are not. The virus remains either way. What stage of technological development correlates with civilizational disappearance in Hanson's statistical model? What is the complete active expression profile of shared human Neanderthal ERV sequences under stress conditions? And if the filter is ahead, how close are we? John von Neumann delivered the lectures on self-reproducing automata between 1948 and 1949, published posthumously in 1966. The argument: any system capable of self-reproduction must contain a complete description of itself. The description must be readable by the system. The system must be capable of copying the description and passing it to the next generation. Life does this through DNA. The genome is the description. The cellular machinery is the reader and copier. But the description does not explain its own origin. The first self-reproducing system required a description of itself before it existed to write that description. Von Neumann identified this as the foundational problem. It remains unsolved. If life is fundamentally an information system using matter as substrate, and if information about Earth's life has been radiating into space since the first organisms produced electromagnetic signatures, then any civilization capable of detecting that signal has known about us for longer than we have known about them. The signal travels at the speed of light. It does not stop. It does not ask permission. Every radar system pointed at the sky, every spectroscopic scan for biosignatures, every radio transmission we send searching for contact, these are additional signals added to the ones already traveling. To a civilization that reads biology as information, the coordinates of Earth and the full chemical description of its life forms are already in transit. We are not deciding whether to announce ourselves. We announced ourselves the moment the first organism metabolized. In November 2018, He Jiankui announced at a genome editing summit in Hong Kong that he had produced the first gene-edited human children. CRISPR had been used to modify embryos before implantation. The off-target effects, unintended edits at other genomic locations, were not fully characterized. The children exist. Their edited genomes will be inherited by their children. We edited the human germline before we could read the full consequences. We did it with a tool that makes cuts at locations it was not directed to cut. We do not know everything that was changed. The changes will propagate. What is the complete off-target edit profile in the He Jiankui subjects? And what will those edits express across generations? At what distance has Earth's electromagnetic biological signature been detectable to a civilization with adequate instruments? And if we are already visible, how long have we been visible? NASA's working definition of life, adopted in 1994. A self-sustaining chemical system capable of Darwinian evolution. Fire is self-sustaining and chemical. A computer virus evolves under selection pressure. A mule is biological and alive and satisfies neither condition. The definition was not replaced because nothing better was produced. Mark Badow and Carol Cleland compiled the complete history of the attempt in The Nature of Life, published by Cambridge University Press in 2010. Every definition proposed across 150 years of biology fails under pressure from at least one real counterexample. The field did not converge on a solution. It documented the failure. This is not a gap waiting to be filled. It is a structural problem with the question. Life is the thing doing the defining. The instrument and the object of measurement are the same system. The definition collapses because the definer cannot step outside itself far enough to describe what it is from the outside. You have been alive for every second of your existence, and you do not know what that means physically. Science has been studying life for 150 years and cannot produce a complete account of what distinguishes it from non-life. Every instrument built to detect or protect life was built by a system that does not fully understand what it is building for. Every threat described in the preceding 40 bits. The prion that needs no code, the viroidal fragment that commandeers your cellular machinery, the mirror organism your immune system cannot see, the ancient sequence sleeping in 8% of your genome, the autocatalytic chemistry assembling in urban infrastructure, the material arriving annually from the outer solar system, all of these exist in the gap between what life is and what we can define it to be. The gap is not closing. The research is not converging. The definitions keep failing. I have spent a long time looking at this from different angles. The conclusion I keep reaching is not that we are ignorant in ways that will eventually be resolved. It is that we are ignorant in ways that are structural. The thing we are trying to understand is the thing doing the understanding. That recursion does not resolve. You are alive right now. Something is maintaining the improbable local order of your body against the thermodynamic pressure of the universe. That something has no complete physical explanation. It has been doing this without your awareness or consent for your entire existence. It will stop. You will not know exactly why, any more than science knows exactly how it started. The most disturbing thing in biology is not any specific threat. It is that the system keeping you alive is one we have never fully described, and we have been trying for 150 years. What would a complete physical account of life require that current science does not have? What does the persistent failure of every definition imply about the relationship between the definer and the defined? And what does it mean to build medicine, biosafety, and planetary protection on a foundation that cannot define what it is protecting?